Chapter 9, Cell Reproduction. So why are cells so small? We talked about this before we got into mitosis and cell reproduction and cell division. There are two main reasons that why cells remain small. Number one is as they get larger, their volume increases much quicker than their surface area. It can be a positive thing to have an increased surface area because I can have more area for chemical reactions, etc., to occur. It's kind of like when we have cellular respiration going on in the mitochondria, it goes on in the cristia, that is the folded areas inside the mitochondria that allows for more surface area for that. But as the surface area increases, so does the volume, and the volume increases at a higher rate. And as the volume increases, two things happen. It makes it much more difficult and efficient to get what is needed into the cell and what is needed to get out of the cell, So that's one of the reasons. The second reason that cells are so small is that as that volume increases, it also makes it much more difficult for the DNA to give its instructions to all of the other parts of the cell and to make things happen the way they're supposed to happen. So chromosomes and eukaryotic and prokaryotic cells. As we know, eukaryotic cells are much more complex in terms of their chromosomes and their DNA. Vocabulary term gene is it's a small section of your DNA that codes for a protein. Again, remember all the important functions that proteins have and how important they are, and we'll come back to that later. Eukaryotic cells have about 100,000 genes per cell, so we're talking about per every individual cell, where prokaryotic cells have about 3,000 genes per cell. Chromosomes are long strands of DNA, and they are found in the nucleus. Remember, back to our prefixes, chroma means color and soma means body. Here's a picture where you can see where everything kind of fits into it. You have your DNA that goes up into your genes, and then they're into the chromosomes, and they make part of that chromosome that are then in the nucleus. When the cell is in the process of dividing, you can clearly see the chromosomes using even light microscopy, which is the same type of thing that we use. Other times, chromosomes exist as loose, diffuse mass called chromatin with an N, and that's when they're in their nucleus and they're just hanging out doing the dance of the chromatin, and that's tin, T-I-N, and that's when they're loose and diffuse. You need to know that for your test. Each chromosome is one long molecule of DNA. It's important to remember. And how many chromosomes are present in a cell depends on the species of that organism. So, for example, dogs have a certain number of chromosomes, humans have a certain number of chromosomes, turtles have a certain number of chromosomes, and they're unique to that species. And that's important for you to know because you'll need to know that on your test. Chromosome duplication, here's some vocabulary that you need to know and understand for it. Some of it went along with our vocabulary quiz. First of all, chromosome is one unduplicated strand of DNA. Sister chromatid with the D is when you have two copies of your chromosomes. It's after the chromosomes replicate at that end of interface there, and they attach to the other copy by the centromere. They make that X sign. The centromere is the region where the two of those sister chromatin are joined together. We're in the middle of that X. If you look at this picture here, you can see the X and you can see where the centromere is. You can see where the DNA is all stranded up into those two chromatins, and they're exact copies of each other. So why do you think that we need to make two copies, and that if we make those two copies that they are joined together like the sister chromatin are? Because if not, it would be like a big mesh and ball of socks here that come out of your dryer. You don't know what matches up with what. It takes time to go through and figure all that out, and some can get lost. Remember, you throw your socks in the dryer and they can get lost. So your sister chromatin are matched up together so that you don't lose them and that they stay together. So just like the pile of socks. So let's talk a little bit about the cell cycle. The cell cycle is the sequence of events of a cell's life. It is from the time that a cell divides into two new cells to the time where those two new cells divide. So cell cycle is the whole gamut of the life of the cell and the division of the cell, and then it goes back to the life of the cells. So there are two major parts to the cell cycle, and you need to know both of these. We first have interphase. Interphase is about 90% of the cell's life, and this is where it does its normal everyday things. It grows, the chromosomes replicate near the end when it's getting ready for division. This is a high metabolical activity going on. There's a lot of chemical reactions occurring, and this is where cells spend most of their life. Here's an example of looking into a microscope and seeing interphase. You can see where your nucleus is, your nucleolus. Usually you just see the nucleus, but you don't see a lot of separation. You can't really see the chromosomes at this phase. Next is what we have cell division, and cell division includes two major processes. The first process being mitosis, which is PMAT, prophase, metaphase, anaphase, and telophase. This is when your nucleus divides. It divides evenly and distributes to two daughter nuclei. After the nucleus divides into two nuclei, then the cytoplasm has to divide, and we call that cytokinesis, all of which you need to know for your tests. So, the phases of mitosis are, number one, prophase. Remember, the dog pees on the mat. PMAT. Prophase, metaphase, anaphase, telophase. That's the order. During prophase, that chromatin condenses and it gets really thick and it thickens into chromosomes. Remember, we said that during cell division we can see these chromosomes under the microscope. We don't see them in interphase. The nuclear envelope, which is the same as the nuclear membrane, begins to disappear. Spindles start to form, and they're composed of microtubules, and these form from the centrioles. And again, the centrioles develop during the end of interphase. During prophase, the spindles start to come out of those centrioles, and they flip to each end of the cell. So the two centrioles aren't together anymore. One's at one end of the cell, and the other's at the other end of the cell, and they have spindles coming out of them. So if you look at this slide here, it gives you a great example of what the prophase looks like. And you start to see the chromosomes start to thicken up. Next is metaphase. And during metaphase, what happens is, sorry about that guys, there's prophase again. Metaphase, what happens is all the chromosomes line up in the middle. We have our X's, sister chromatin, and they are going to line up in the middle like this. And the spindles are going to come and attach to one of each one of those. So they're going to line up in the middle, and that's what it looks like. You're going to need to be able to recognize these different looks by looking at actual pictures on the test. Next is anaphase. And in anaphase, the centromeres divide, which means those sister chromatin separate. So the spindles pull the sister chromatins in opposite sides of the cell. Again, we know that ana means to move away, to go away. So they're moving away from each other. This is what anaphase looks like in a cell. They start to pull away from each other, where they'll be close together in early and farther apart in late. Next is telophase. We know that tele means distance or end. The chromosomes are going to gather at the opposite ends or sides of the cell, and this is also near the end of cell division. The new nuclear membranes start to form around each group, so I kind of have the big cell with the two new nuclei in them, kind of like when we had the big cell membrane out on the floor when we did our activity with our pool noodles. And this is what it looks like here. Now remember, and you need to know this for your test, in the formation of cytokinesis, you're going to start having a cell plate form in animal cells because that helps form the cell wall. In cytokinesis of animal cells, they just kind of pinch off like we did in our activity that we did in the hallway. So what is it that tells the cell it's time to move into the next phase of the cell cycle? What is it that controls that? And it's controlled by an interaction of proteins called cyclin, which is one of our vocab terms this week, and enzymes. Again, the importance of proteins show up again. What happens when a cell divides uncontrollably is that we get cancer. And cancer is a malignant growth resulting from uncontrolled cell division, and you need to know that for your test. And it is caused by environmental factors and changes in your enzyme production. Again, here we go, enzymes, which are made of proteins. So what controls that enzyme production? Number one, your genes do. So this is the genetic component. A gene, again, from our vocabulary terms, is a segment of DNA that controls production of a protein. Cancer can be genetic because these genes are what controls the enzyme production, which controls the cell production. There are also environmental factors such as smoking, tobacco and tobacco use, smoking and chewing. That gives you a lot of different cancers, not just cancers, lung cancer. Air and water pollution, exposure to ultra radiation from the sun, especially going into tanning booths. It's very bad for you. It increases your chances of getting melanoma dramatically by just going into a tanning booth once. So stay away from those tanning booths. Also viral infections. You know, viruses live by taking over our DNA. And by taking over our DNA, they can disrupt this enzyme production. So what can we do to prevent cancer? Diet is very important. Low-fat, high-fiber diet, you need to know that for the test. Kinds of foods you want to eat, fruit, grains, vegetables. You want to make sure you get your vitamins, minerals, especially getting vitamins and minerals from your foods, not just from vitamin supplements. That's all very important. Also, stay away from smoking, tobacco use of any kind, and excess sun. That will all help with the prevention of cancer. Please also make sure that you look at the cancer article from the National Geographic article that we looked at. Please make sure that you look at that so that you know your interesting facts about that, because that will probably, something about that will probably show up on the test. Also, the kind of questions that are going to be asked on the test that have to do with the extended responses may look something like name and explain 2 of the factors that can limit cell size. Something might look like, why is it necessary for a cell's chromosomes to be distributed into its daughter cells in such a precise manner? So why do we have to have that division of the nucleus be so precise and organized for that? So those are the type of things that you will need to know in terms of your short answers. Also, you're going to have to be able to look at pictures and be able to tell what the different phases are of the cells, and maybe look at pictures and put them in order and be able to tell what's prophase, metaphase, anaphase, telophase. So please be able to recognize those in pictures, kind of like we looked at in the root cell. Know your phases. That will be very, very important for you. Okay, I hope this helps you with the test. Good luck.